Hello, everyone. Thank you for joining our webinar today. My name is Helly Pimentel. I'm a product manager here at ACD. Today, I'll provide you with a sneak peek into a very exciting new product that will be released next month. If you have any questions throughout the webinar, please use the chat function to the right of the WebEx window and address your questions to all panelists. My colleagues will address your questions in real time. At the end of the webinar, if you're available to stay behind for a few more minutes, I'll try to address some live questions as well. In the event we have not addressed your questions, we'll follow up with you offline after the webinar. On the agenda for today, I'll first go over ACD existing product portfolio. Then I'll switch gear and introduce you to our new exciting RNA ISH assay to detect small RNAs. I'll be sharing some data to demonstrate the new assay's performance, along with some applications data. Towards the end of the webinar, I'll summarize today's presentation and conclude with a Q&A session to address some of your questions live. To begin, ACD's core technology is the RNAscope technology. RNAscope is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells or tissues with morphological context at the single-cell resolution. The RNAscope technology consists of three parts. First, a unique target probe that ACD designs against a sequence of interest, and a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots for quantitative analysis. The assay allows for spatial mapping of mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, and point mutations in cells and intact tissues, all which can be visualized with either fluorescent or chromogenic labels. In addition, the assay can be performed on a wide variety of tissue samples, including FFPE tissues, fresh frozen or fixed frozen tissues, and culture cells. ACD's technology includes two unique assays to detect RNA in tissue context at single molecule sensitivity with single cell resolution. First, RNAscope is designed to detect any mRNAs or non-coding RNAs that are greater than 300 nucleotides in length. RNAscope probes are designed in about 20 ZZ probe pairs. On the other hand, the BaseScope platform uses probes that are designed in 1 to 3 ZZ probe pairs to specifically support the applications of splice variants and highly homologous sequences and point mutations. The RNAscope workflow is very similar to IHC if you're familiar with tissue-based protein assays, so you'll find it quite intuitive to adopt this workflow. So first, we start with properly permeabilized cells or tissues, then hybridize the probe to the target RNA of interest. Next, with addition to the proprietary amplification system in the form of detection reagents, which will allow for the visualization of a single RNA molecule in the form of dots, and which will enable us to quantify the level of gene expression. Our products cater to each of these steps, providing ready-to-use reagents for most cases. To design a specific probe for your target of interest, ACD requests you to provide us with the accession number or the exact sequence of the target. You could even indicate your region of interest for targeting with start or stop positions. Our bioinformatics scientists will channel this information through our proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm will evaluate the sequence composition of each oligo in the pool to ensure that no cross-hybridization with non-target RNA will occur under stringent RNAscope conditions. The highlight of the RNAscope technology is in our probe design strategy. We can design probes for virtually any species and have already done so in over 200 species, covering most applications highlighted in our website. The majority of our probes are designed in human and mouse. On our website, you'll be able to find the applications areas to demonstrate how these probes are used in the most common applications, including infectious diseases, cancer, neuroscience, and gene therapy. We've also added support for signaling pathways such as Wnt or DES and indicated probes we have available within that pathway. This slide summarizes all the assays that are currently available from ACD. We have chromogenic singleplex brown or red assays, and also duplex assays on both the manual and automated platforms. The multiplex fluorescent assays allow for the detection of up to 4 targets simultaneously in a single tissue section. The major difference between the two fluorescent assays is that the V2 assay uses fluorophores from Macquarie Biosciences for signal visualization and provides a high signal-to-noise ratio, whereas the fluorescent multiplex assay is an all-in-one kit. The V2 fluorescent assay is also available on the Leica automation platform. For higher plexing, we have the RNA-scope high-plex assay, which will enable the detection of up to 12 targets simultaneously in a single tissue section. Currently, the high-plex assay is available in the manual format. If your target of interest is shorter than 300 nucleotides and studying splice variants, short or highly homologous sequences, or point mutations, then the BaseScope assays would be the right choice for you. Currently, the BaseScope singleplex assay is available on all platforms, while the duplex assay is available on the manual platform. Next, expanding our product portfolio, not only do we have assays to detect mRNAs, long non-coding RNAs, short targets, point mutation splice variants, but we'll be releasing a new assay next month to detect small RNAs, including antisense oligos, microRNAs, and small interfering RNAs. So what is this new assay? Before we go into the details of the new assay, we'll look at the significance of detecting small RNA species. Detection of these smaller RNA species can have significant pathological relevance. Specific and sensitive detection of small RNA targets, such as ASOs, microRNAs, and siRNAs, can be very challenging. These require probes that are designed against very specific short sequences on the target of interest. For example, ASO-mediated therapy is a new form of RNA interference solution to inhibit specific target genes. The detection of ASOs requires highly sensitive technology to evaluate the specificity of ASO-mediated therapies and also to study gene expression after the delivery of the ASO therapy. Next, microRNAs can influence not only a single gene, but the entire cellular pathways or processes. So visual detection of microRNAs are very important to examine subcellular localization and gene expression. Last but not least, siRNAs are one of the most promising types of RNA-based therapeutic approaches to silence target RNAs. As a result, the ability to validate siRNA therapy is very important to assess the specificity and efficacy of the siRNA therapy to avoid any off-target effects. Attempts at detecting small RNAs have relied really heavily on the qPCR, small RNA sequencing, and microarray approaches. While these methods deliver both expression levels, they do not provide detailed spatial information for gene expression. Additionally, existing small RNA ISH technology suffers from poor reproducibility and sensitivity and also requires extensive optimization, therefore consuming substantial quantities of specimen samples. So this emphasizes the need for a technology that can reliably detect cell type-specific small RNA species in a tissue while maintaining high detection sensitivity and specificity. So to address customers' needs, we're delighted to share with you that ACD has developed a new RNA in situ hybridization assay called the miRNA-scope assay to allow for the detection of small RNAs with spatial and morphological information at single-cell resolution. The miRNA-scope assay will enable the detection of short RNA sequences, including ASOs, microRNAs, and siRNAs, supporting popular applications such as cancer, infectious diseases, neuroscience, and biopharma therapeutics. Additionally, this new assay leverages ACD's core amplification and probe design capabilities to enable the detection of RNA molecules at single-cell resolution, while also improving the detection of short target RNA with ease of data analysis. To enable further insights into your research, this new assay is also compatible with EDGE-C, detecting small RNA and protein simultaneously on the same slide. Next, we'll focus on the miRNA-scope assay workflow. The new miRNA-scope assay workflow is very similar to the RNA-scope singleplex workflow for those of you who are familiar with our technology. First, the cells, the tissues that are bound to a slide is permeabilized using ACD's ready-to-use pretreatment reagent. The new assay supports a variety of sample types, including FFPE, fresh frozen, and fixed frozen tissues, and culture cells. Then the small RNA target probes are hybridized to the target RNA. Currently, the microRNA scope assay allows for a single RNA target detection. The hybridization step is universal across all targets, meaning further probe optimization is not required. After probe hybridization, the target probes will be hybridized to ACD's modified branch signal amplification tree, consisting of amplifiers and label probes to provide high sensitivity and specificity. After the colors have been developed, the small RNA molecule signal can be seen as a red chromogen deposit using bright field microscopy. I would also like to mention that the fast red chromogen can also fluoresce similar to the ADO 550 dye under a fluorescent microscope. The slides can then be also scanned for further qualitative or quantitative analysis. The entire singleplex assay workflow can be done either manually or fully automated using the Leica systems. Next, we'll focus on the applications of the new microRNA scope assay. Again, this new assay will enable the detection of small RNAs such as ASOs, microRNAs, pRNAs, small hairpin RNAs, and siRNAs. It also supports the sequential ISH detection of RNA and protein on the same tissue slide. Major research segments for this new assay includes basic science with the application of neuroscience, stem cells, and developmental biology. Next major research is infectious diseases in the study of inflammation and immune cell regulation. Following that, biopharma therapeutics is an important segment studying delivery chemistry, biodistribution, safety and toxicity, and drug stability. Last but not least, the new assay will support the study of cancer and clinical research around tumor biology, immuno-oncology, and pathology. Overall, these applications require a highly sensitive and specific solution to study small RNA expression. And that is why in the next few slides, I'll show you some of our microRNA scope assay data to demonstrate the assay performance. First, I'll show you some data demonstrating assay specificity with some of them validating published results. Here I'm showing is a publication using Northern blot analysis to show microRNA 124 expression in different mouse organs. The analysis showed that microRNA 124 is predominantly expressed in the brain tissue, and it's also one of the most abundantly expressed microRNAs in the brain. And on this slide is our internal data demonstrating robust assay performance across tissues with the microRNA scope 124 probe. In here, we observe high microRNA 124 detection in FFP normal adult mouse, rat, and human brain tissues. On the other hand, the mouse liver is a known negative tissue, here showing no microRNA 124 detected. This data demonstrates our new microRNA scope assay specificity as well as concordance with existing assays such as the Northern blot analysis from the previous publications that I just showed you. This slide is also data showing detection of microRNA 124, but in FFPE mouse embryo tissue. As you can see here, microRNA 124 is detected predominantly in the mouse brain, but absent in other tissues such as the lung, liver, and kidney samples, demonstrating the specificity of the probe and further confirming the Northern blot analysis. Additionally, we also use a scramble probe to further demonstrate the specificity of our assay. Here you can see that the scramble probe is very clean in the mouse embryo tissue with no background. Another example that is shown here is the detection of microRNA 140 in mouse embryo cartilage tissues. Existing publications indicate that this microRNA is specifically expressed in cartilage tissues. Our microRNA scope assay demonstrates that indeed microRNA 140 is detected specifically in mouse cartilage tissues such as the digits, somite, and ear. We also stain the same mouse cartilage tissues with our scramble probe to further demonstrate the specificity of the target probe, where you see no background is detected. we have data showing the detection of microRNA 122 in different species samples. MicroRNA 122 shown here is detected in high levels in liver samples from mouse, rats, and human species. To demonstrate specificity of our assay, microRNA 124 was also stained in a negative control tissue, the mouse kidney, here showing no detection of the target. Scramble flow also shows no background detected here. The last slide to demonstrate our assay specificity is looking at the detection of microRNA 138 in mouse brain tissues. This specific microRNA is known to be expressed in Purkinje cells in the brain regions such as the cerebellum and hippocampus. What this data shows us is that our microRNA scope assay offers highly specific detection of targets in specialized cells of the brain. Next, I'll show you a few slides to demonstrate sensitivity of our new microRNA scope assay. Here's an image showing the detection of the outer nuclear layer specific microRNA 182 in FFP mouse eye sample. As expected from published data, negligible microRNA 182 is also detected in the inner nuclear layer and the ganglion cell layer. This demonstrates that our new assay is capable of detecting microRNAs at different expression levels in subcellular types. Next, shown in this slide is a publication showing the detection of microRNA 145 being selectively expressed in the vascular smooth muscle cells using qRT-PCR and northern blot analysis. Our internal data using the new microRNA scope assay validates the published data with the qPCR and northern blot analysis from the previous slide. As you can see here, microRNA 145 is detected specifically in the vascular smooth muscle cells in mouse lung, intestine, and kidney samples. Our assay also shows you spatial information that other microarray assays do not show. Last but not least, visual detection of microRNAs can be very challenging sometimes due to their low level expression. Our microRNA scope assay has successfully demonstrated a specific detection of lower expressors as well. Here are examples of lower microRNA expressors with the detection of microRNA 124 in the mouse kidney sample, microRNA 31 in mouse intestine tissue, and one of the most popular microRNA family members, +7A in mouse lung tissue, using our new microRNA scope assay. In the next slide, I'll talk about our recommended assay controls for the microRNA scope assay. The microRNA scope assay to detect small RNAs will offer a universal positive and negative control probes. Strong nuclear U6 signal is observed in a variety of tissues, including human and mouse tissues, and HeLa in 3T3 cell pellets. That is shown here on the first row. A universal scramble probe is recommended as an assay control to demonstrate the specificity of the assay across species and samples, as shown in the bottom row. Next, I'll show some data using our microRNA scope assay for the detection of ASOs and siRNAs. The detection of these small RNAs are very important, as previously mentioned, for validating delivery chemistries, biodistribution, safety and toxicity, short-term and long-term stability for your therapeutic solution. Here's an example of a customer using our microRNA scope assay to validate their ASO-mediated therapy. The vehicle tissues here are used as a control sample, and the treated tissues are where the ASO compound is delivered. As you can see, the ASO target is detected in high levels and only in the ASO-treated mouse cardiac and quad muscle samples. The control vehicle sample shows no detection of the ASO target. This data will help the customer to conclude that this was a successful delivery of their ASO therapy. In this next example, our customer has generated 2 different siRNA compounds to determine the best solution for their siRNA therapy. In the provided samples, we successfully detected the siRNA number 1 target in the siRNA number 1 delivered sample, and not in the semi-negative control nor the siRNA number 2 delivered control sample. Using our microRNA scope assay, the customer was able to study the biodistribution of the siRNA number 1 target delivered in the mouse liver tissue as well. Next, we'll focus on the microRNA assay ISH compatibility. A great way to further characterize cellular gene expression is to combine ISH with IHC on the same slide to simultaneously detect RNA and protein. Because of the similar workflow between ISH and IHC, including sample fixation, pretreatment, pro-hybridization, signal detection, and data analysis, ISH and IHC are ideal to combine into one workflow in which our ISH assay is performed first, followed by the IHC assay. What we refer to as a sequential ISH-IHC here can be used to characterize cell type-specific expression, identify origin of secreted proteins, visualize cell surface markers with RNA of interest, visualize RNA binding proteins and their target RNA, and also dissect regulation of gene expression. Here's an example of sequential ISH-IHC staining performed on human cervical cancer tissue. The mRNA-specific microRNA 223 and T-cell marker CD3 were detected on the same tissue section. Our ISH and IHC staining shows mutually exclusive localization of these two targets. Here's another example of sequential ISH-IHC staining performed on human ovarian cancer tissues. Again, showing the detection of the mRNA-specific microRNA 223 and T-cell marker CD3 on the same tissue section, showing mutually exclusive localization of the two targets again. So to summarize today's topic, our new microRNA assay is a robust RNA in situ hybridization assay, enabling the detection of small RNAs in tissues with spatial and morphological context at single-cell resolution. Key applications of our new assay include ASOs, microRNAs, siRNAs, and other short RNA sequences that are 17 to 50 nucleotides. The new microRNA assay will be a single-plex chromogenic fast red assay, which is compatible with the manual platform or the fully automated Leica platform. We understand that challenges of many of us are facing during the pandemic. However, research must go on. That's why our services team are also here to assist you with moving your research forward. Our pharma assay services are fully operational and could help you advance your research in the detection of ASOs, microRNAs, and siRNAs if you'd like early access to our new microRNA assay. In fact, we have successfully completed multiple pharma assay services for our customer using the new assay to detect their small RNA of interest. Here's an anonymous testimonial from one of the leading biopharma companies in the US who completed the siRNA study through our pharma assay services team. This is what it read. They had a great experience using ACD's microRNA assay in their compound screening efforts. Their technique is incredibly specific and the results are very easy to understand. And they also mentioned that ACD generated impeccable tissue stains. The official commercial release of this new microRNA product is scheduled for the end of July. Please feel free to contact us with any questions you may have with the following contact information. To conclude the webinar, I'll address some live questions from the chat box. For those you exiting the webinar, we thank you for joining the webinar today, and I hope you're excited about upcoming new products. So, Rhonda, could you please read me some live questions from the audience?
Thank you, Helly. That was an excellent presentation. Thank you so much. And for those of you that joined a little late, this was recorded, and so we'll make sure that the recording gets out to you shortly in the next couple of days after today. We did get a couple of questions, Helly, so let me read them through for you. The first one was, how do you determine specificity of the target probe?
Thank you, Rhonda. So to determine the specificity of the target probes, we recommend using our universal scramble probe to evaluate the specificity of the target probe. Optionally, we also could design a specific scramble sequence to a target sequence to evaluate specificity. For ASO and siRNA target probes, we recommend your study to include a control or untreated sample to evaluate the target specificity.
Thank you, Helly. That was great. Second question I have here is, can the miRNAscope Pros be used with the existing RNAscope or BaseScope assays?
That's a great question. The miRNAscope Pros must be used together with the miRNAscope detection reagents. The use of the miRNAscope probes with the RNAscope or BaseScope assays will result in suboptimal results. At this point, detection of mRNAs and microRNAs on the same slide is not yet available.
Okay, good to know. Thank you for pointing that out. Another question here is the miRNAscope assay protocol the same as the RNAscope or BaseScope assay?
The miRNAscope assay protocol is very similar to our RNAscope 2.5 singleplex red assay. The manual assay protocol has an extra post-fixation step that is required for optimal assay performance.
Great, thank you. And then just in addition to that, how about the tissue types that it's been designed for? Is that different to our standard assays?
The tissue types that are compatible for the miRNAscope assay are the same as for RNAscope and BaseScope assays. So FFP tissues, fixed frozen or fresh frozen tissues, as well as culture cells. If you're already using the same tissue samples with our RNAscope or BaseScope assays and want to use the same tissue samples for miRNAscope assay, you can follow the previously established pretreatment conditions. So then it can be used for the same pretreatment conditions for the miRNAscope assay. For new users, if you're using, you never used our assay previously, we have a set of pretreatment recommendations as well, which you can follow in the user manual.
Great, thank you. I was actually going to follow up without pre-treatment. No worries. That sounds really great. So we're really excited about this product coming out, and you said mid to end July. So that's really exciting, and I know there's a lot of buzz going on on our social media channels as well. So for those attendees want to learn a little more, we'll have some more information coming out. Keep an eye on our website for more information, and look forward to us sending out an email with this recording in it as well. With that, that's the end of the questions that I have. Thank you very much for joining us today, everybody, and thank you so much for your presentation, Helly. We look forward to next month. Okay, thank you very much.
Thank you, everyone.